Welcome you for the CME on hypoglycemia. Needless to emphasize that hypoglycemia is the most important problem that we face when we treat patients of diabetes. In this CME, we are going to look at the pathogenesis, the counter-regulatory mechanisms of keeping the blood glucose in the very narrow range of 70 to 100 milligrams, and the diagnosis of hypoglycemia, the different types of hypoglycemia, and of course the treatment of hypoglycemia. As usual, this CME is also available on our website www.drsharma.in. In this website you can find several other CMEs of around 140 different CME topics of interest for free download. I request you to go to the website and take advantage of these different CME presentations. So let us now proceed to understand hypoglycemia. Before we go into hypoglycemia, we need to understand the glucose homeostasis. So, the blood sugar or blood glucose is kept in a very narrow range of 70 milligrams to 110 milligrams in the fasting state, with a slight spike after the postprandial state up to 140-150 milligrams. How is this possible? This is because of two important hormones, the insulin and the glucagon. So let us start with a scenario where the patient's or person's blood sugar is on the lower side, less than 70. Immediately the alpha cells of the pancreas release glucagon. This glucagon stimulates glycogen breakdown and also the production of new glucose called neoglucogenesis or gluconeogenesis. With the breakdown of glycogen, new glucose is available into the system and new glucose is manufactured from the precursors by gluconeogenesis. Both these will increase the blood sugar. Of course, when the blood sugar is low, there will be also stimulation of the hunger, and we would take some food which contains the carbohydrate, which will be converted into glucose, so much so the blood sugar is brought to a higher level or normal level. Supposing, look at a scenario where the blood sugar is higher than normal, let's say 150, 200, like that. Immediately the beta cells of the pancreas, instead of the alpha cells there, now it is the beta cells of the pancreas that are stimulated, and they produce insulin, which in turn stimulates glucose uptake by the peripheral tissues, and also this movement of glucose, inward movement of peripheral tissues, reduces the blood sugar to normal levels. So in between meals, this is how the blood sugar is maintained at a lower stage of around 70 to 110 milliequivalents. So this is in nutshell the glucose homeostasis as we see here with the glucagon and the insulin. Whenever there is low blood glucose, glucagon comes into picture. Whenever there is higher blood glucose, insulin comes into picture. Glucagon produces input of more glucose into the system. Insulin removes the glucose from the system into the tissues. So this is glucose homeostasis. Whenever we have excess of glucose in the system, it will be stored as a fixed deposit in the liver, in the muscle, and to some extent in other tissues, adipose tissues, as glycogen. This process is called synthesis of glycogen or glycogen synthesis. Glycogen is the storage molecule or storage form of energy. Glucose is converted into a complex carbohydrate called glycogen and stored for needs. Whenever there is need, whenever the blood sugar falls, before we take some food to raise the blood glucose, this glycogen that we have stored in the liver or the muscle is broken down, and this will be able to keep up the blood sugar levels and keep us going until around 8 hours of time before we start eating further meal or snacks. So in general, up to 8 hours we will not have any problem as far as the maintenance of blood glucose is concerned. That is because of stored glycogen is converted into glucose by a process known as glycogenolysis. Of course, this 8 hours will be shortened whenever we exercise, whenever we are starving, whenever there is illness, and of course most importantly, whenever the patient is an alcoholic. Because alcoholics, the glycogen storage in the liver is much lower, and so much so, in these conditions like exercise, starvation, illness and alcohol increase, alcohol intake, in all these conditions, the glycogenolysis is increased, so much so, the ability to withstand fasting will be reduced from 8 hours to 4 hours or even less. What is gluconeogenesis? Gluconeogenesis is, neo means new, genesis is production, so new production of glucose. That means glucose, even if you do not consume glucose, glucose can be synthesized from the precursors like the fatty acids, like the proteins, amino acids, protein breakdown products, amino acids. So from the amino acids and from the fatty acids, glucose being synthesized is called gluconeogenesis. This occurs mainly in the liver and the kidney, and muscle and adipose tissue and kidney supply the fuels. The fuels are the fatty acids from the adipose tissue, and the muscle gives the protein, and the kidney also gives some of these precursors, and all these together are combined to form new glucose. Remember, brain can use only glucose and has got very little glycogen with it, so it cannot withstand the hypoglycemia for a long time. It can sustain only a few minutes without glucose. Luckily, brain does not need insulin for the transport of glucose because in all other tissues, when the glucose has to enter the cell, they require a specific transporter called the glut, and these gluts are under the influence of insulin. But in the brain, the glut is there, but the type of the glut that is there in the brain does not require, a glucose transporter in the brain does not require insulin. That is the reason when we treat hypoglycemia, we don't require to give insulin. Otherwise, if brain were to need insulin, we would have treated every patient of hypoglycemia with glucose along with insulin. So that is the situation as far as the homeostasis is concerned.